Have you ever wondered what would happen if you gave an octopus MDMA? No? Me neither, but some scientists decided they wanted to find out. The octopus, class Cephalopoda, phylum Mollusca, a soft-bodied creature with 8 limbs, a shark beak, and extreme intellect. They can slip out of crab traps in aquariums, open jars, or use their insects to escape predators. Octopi are known for being solitary animals. They swim alone, live alone, and hunt alone. However, this changes when mating season comes around. This makes octopi an important model for testing social interactions, as they possess the capability to be social but do not normally exhibit that behavior. MDMA has been found in humans to act on the 6th transmembrane domain of the serotonin transporter, SERT, encoded by the SLC6A4 gene. Scientists analyzed the sequenced genomes of a number of species looking for this particular gene, or similar analogs to this gene, and found that a number of social and asocial animals had some form of this gene present, but eusocial animals like ants and bees lacked this gene. Interestingly enough, despite 500 million years of divergent evolution and how different the anatomy of our brains are, O. bimaculoidus, or the California two-spot octopus, shares a copy of this gene with us, with the parts of the gene coding for the combined binding site for serotonin and MDMA having a 100% match with humans along the 4 base pairs that part occupies. This means they would be able to experience the effects of MDMA.
In terms of how the experiments were set up, a glass aquarium was separated into 3 equally sized chambers with a captive octopus, or a male or female social object. Inside a perforated container in the right chamber, the test O bimaculoids, or the California two-spot octopuses, were originally placed in the middle and the objects in the left. Each tested animal subject spent a maximum time limit of 30 minutes freely roaming in the aquarium. 2 experiments were run in the aquarium. Experiment 1 tested for preference and interaction between an intimate object and a social object octopus, both male and female. This was done by placing male and female octopi in the center chamber and monitoring time spent in different chambers over 30 minutes. Both male and female octopi were found to spend more time in the chamber with female social objects, whereas when the social object was male, they spent more time in the object chamber. According to Figure B, the average time spent in each of the 3 chambers is along the y-axis, with the blue bar representing the object chamber, pink the starting chamber, and yellow being the social chamber for both male and female social objects along the x-axis. The average results demonstrated that there is an aversion to interaction with male octopi for both genders, and that octopi preferred interaction with female octopi. Thus, the best test to determine if MDMA increased the social interaction was in the aversion scenario with a male social object. In the second experiment, a pre-observation was conducted for 4 of the California two-spotted octopuses, with 3 being male and 1 female. During this, no MDMA was given to the test subjects, and they each were observed for the 30-minute time period in the 3-chambered aquarium with a male social object. After these observations, the test subjects were given a 5 to 24-hour break, after which the MDMA was administered by mode of a 10-minute soak in MDMA-treated water, followed by a 20-minute saline bath, since research shows that the most prominent effects of MDMA begin to be seen between 30 to 60 minutes after ingestion. Each of the 4 tested octopuses were given a submergent dose range between 0.5 to 0.005 milligrams of MDMA per kilogram of the weight of the octopuses. At that point, they were put into the test chamber and monitored for 30 minutes. The timeline of this process is represented by Figure A. The scientists found that in the post-MDMA trial, the octopi spent significantly more time in the social object chamber and initiated much more contact, basically hugging the social object octopus. In the pre-non-MDMA trials, the average time spent in the object chamber was the greatest. Figure C describes this with the y-axis representing the average time spent in each chamber and the blue, pink, and yellow bars being the corresponding chambers for both the pre- and post-experiments along the x-axis.
This shows that octopi exhibit complex neural pathways present in diverse taxa of animals that mediate social interaction via serotonin. It's incredible that such far-removed animals retain copies of the specific genes and gives clues that the SLC6A4 gene plays an important role in social interactions. The mutation, duplication, and analogs of these genes in different animals can be studied to tell the evolutionary story of adapting different social behaviors, and this study helps us understand how those behaviors are biochemically mediated.